Hello, everyone, and welcome to Sustainable Horizons webinar series. My name is Pooja Rao, and I'll be facilitating today's webinar, the topic of which is Introduction to Scientific Visualization. Before we start the webinar, I have a few announcements to make regarding the structure of the webinar. So the webinar is about 55 minutes long, and you're allowed to ask questions during the webinar. So those questions that are asked during the webinar, please use chat for those questions. You can write your questions in the chat, and at various points during the webinar, I will ask speaker these questions on your behalf. If we cannot cover all the questions during the webinar, an additional 15 minutes have been allotted at the end of the webinar to answer any further questions. The speaker is going to be available during that time. Any follow-up questions could be asked via email. Our email is info@shiinstitute.org. We will also have the video recording of the webinar available on our SHI webinar page, the link to which is given at the top of the slide that you're seeing on your screen right now. Additionally, you can have access to your Exceed accounts for up to 30 days after the webinar. So if for some reason you can follow along the exercises in the webinar, you can do it at your own pace and shoot us an email if you have any questions. Very important, there is a feedback form that will be made available to you at the end of the webinar via the chat. Please, please, please fill that out. It's very important for us to hear what you guys have to say about the webinar. It helps us decide the future topics for the webinar and how we can improve the overall experience of the webinar. So please do let us know by filling out the feedback forms. You can keep in touch with us via subscribing to our email list, and we do send out a lot of useful information regarding internships, fellowships, and a lot of other useful stuff. You can follow us on Facebook, Twitter, and LinkedIn as well. So one more thing about using raise your hand option in the additional Q&A session following the webinar. So you can access this raise hand option if you click on the participant menu, the participant option that you're seeing at the bottom of your screen. So if you click on that, that should pop up another window, and at the bottom of that window, the left corner of that window, you should see an option that says raise hand. So if you click on that option, it informs us that you are interested in asking a question, and we might let you ask your question via audio. All right, well, without further ado, I have a very special guest to introduce today before we start the webinar. So Mary Ann Liang, the founder of SHI, is with us here today, and she'll be doing the honors of introducing our speaker for today. So over to you, Mary Ann, and thank you.
Thank you, Pooja, for the kind introduction. I'm delighted to be here today, to have an opportunity to participate in the webinar and to share with you just a little background on Sustainable Horizons Institute in the webinar series. So our mission is to build sustainable and inclusive connections between students, faculty, and professionals in science, technology, engineering, and mathematics, with a special interest in promoting diversity and inclusion. I'm particularly happy to have the honor of introducing Colleen Heidemann, today's webinar presenter. Since I've known Colleen for since 2015, when she participated in the workshop we organized to train and provide assistance for submissions to the technical program of the poster session of Supercomputing 2015 conference. After her participation in the workshop, we helped her obtain a position at Lawrence Berkeley National Laboratory, where she's been a part of the data analytics and visualization group, developing parallel image processing algorithms and analyzing their performance. Colleen also participated in the broader engagement program we organized at the Society of Industrial Applied Mathematics Computational Science and Engineering Conference, and she's been serving on the webinar committee as well. So the webinar committee was formed to provide programming of interest to the high performance computing and computational science and engineering community that she works with. The committee is comprised of volunteers, mostly made up of students or past student benefactors of SHI programs. And this grassroots effort was designed to provide the content that aspiring young students and professionals have said they want and need, and also to provide professional development opportunities for the committee members. I'd like to take a moment to thank all the committee members, which is comprised of Sarah Cooper, who's a genome science PhD student at the University of Tennessee, Knoxville, Anna Sophia Hurst, who's a graduate student in mathematics, Pooja Rao, who you've already heard from, who's a postdoc in aerospace engineering at the University of Illinois, Urbana-Champaign, Wendy Sapp, who's a computational chemist working with Oak Ridge National Laboratory and SHI, and Colleen Heineman, who I'll introduce in more detail in just a moment. So please join me in thanking the entire webinar committee for all their work, because they're just totally awesome and have done a lot to just brainstorm the ideas and put out in all the work to organize the series. So we are interested in engaging community members in our work. So if you're interested in volunteering for the webinar committee or the broader engagement organizing committee, or just any other activity that we're involved in, or if you just have some topics that you'd like to suggest, please let us know. So as you can see, we have been working with Colleen for several years, which makes today a very special day for me. And it has been a delight to watch Colleen grow from a young undergraduate student to a PhD student and a presenter and trainer on high-performance computing and visualization. Another thing that's very rewarding for us at SHI is that Colleen represents not only a real success story of someone who had a dream and worked really hard to find a way to success, she also represents how we at SHI strive to help people through sustained and deliberate program design. Colleen is currently a PhD student at the University of Illinois, Urbana-Champaign, where she utilizes her background in high-performance computing and scientific visualization, visualize data and interactivity through virtual and augmented reality. In addition, she has been an instructor for the Blue Water Student Internship Program for the past 4 years at the National Center for Supercomputing Applications, where she's a member of the Data Exploration Lab. Without further ado, I'll turn it over to Colleen and we'll get the webinar started. Thank you so much.
All right, thank you very much, Marianne. Appreciate it. All right, so we're gonna go ahead and just dive right in to an introduction to scientific visualization. So I'm gonna jump in and sort of just give you guys an outline of what we're gonna talk about here. This is meant to be more interactive with you guys as participants, more like a free dialogue-like discussion. So I might ask some questions throughout the webinar. Please feel free to shout out answers or questions in response to my questions that I ask. Because our goal here is really, we're not going to sit down and teach you step by step how to use a software, step by step how we think you guys should do something in order to visualize your data. The goals are more to give you guys an idea of what visualization is and how it can be utilized, and then also give you enough background information to be able to ask questions about how to visualize certain data sets. So we'll go ahead and start out with what is visualization? And this is kind of a loaded question because pretty much anyone you talk to will have a different answer from another person that you ask. So I'm gonna throw a couple things out there. Commonly used buzzwords that go along with, I guess, the visualization realm in general would be, it's computer graphics. You know, we're just using images. But it's so much more beyond that. What are we actually using the images and computer graphics for? So a big word that I want to throw out there that is very meaningful, and this is just my interpretation of visualization. It's by no means all encompassing, but it's a visual representation. And beyond being a representation, the goal is to provide insight, gain information and knowledge out of whatever data it is that you're actually attempting to visualize. And depending on how you visualize that information, you might gain different insights from each different visualization technique that you use. So why visualization? We've sort of set a definition, and we'll explore that definition a little bit more as we continue throughout this webinar. So this is an exercise I like to do whenever I give this talk. This is a very simple example right here. If we look for all of the boxes here with a number greater than 70, we'll see there's two. We got 75 and 80, both on the right side. And because it's small, that's pretty easy to find. We can do that relatively quickly. But as we increase, we see now there's twice as many squares, and we actually see there's 4 squares with numbers greater than 70, but it takes us a little bit longer to actually count those up and determine which ones are greater than 70. If we continue, we see there's even more squares. It takes us even longer. And if we just continue on with that trend, if we look at this, it's going to take us quite a while to find all of the squares greater than 70, right? So this is just our simple data. Whatever our experiment is, what data it kicks out. We're trying to find some type of meaning from this data. And if we're simply just staring at these numbers, that can be extremely difficult and oftentimes impossible. But what if I do this? I've now put something to associate with within this data. So we see on the side, we've got a color map. 0 is associated with dark blue, 100 is associated with dark red. And we can see in this picture that the numbers that we're looking at are all over the map. So if we want to look for numbers greater than 70, we'll look for things on the color map that we see are probably within the realm of orange and reds. And so we see that there's a big square, or blotch, sorry, right in the middle of our image. And so what is that telling us? That tells us, just off the top of our heads, probably roughly about half of these squares are greater than 70. We don't have an exact number because we haven't sat down to count them all up. We can see it's probably roughly about half or so. And then this representation, this could be telling us all kinds of things. This could be some type of mapping of, we could be looking at terrain heights, something like that. We could be looking at temperatures of locations, anything like that. So it's a specific representation of whatever we say we're going to look at. If we chose to use some other value associated with each of these squares, we might come up with a completely different answer, which means we're getting completely different information out of that, and potentially different knowledge, which could help us advance whatever type of scientific discovery we're trying to make or what we're looking for. So this illustrates exactly what I just said. We're looking at a representation of whatever data it is we have. So chances are our data is going to be spit out just as a bunch of numbers. And if we're doing extremely large scale experiments with high performance computing, doing things in parallel, we can end up with, you know, possibly terabytes of information that just look like this data set here. That's just numbers and numbers. And to the human eye, especially somebody that's not involved in the experiments that you're trying to do, it's pretty useless. You know, I look at these numbers and they mean absolutely nothing to me. I can't even potentially attempt to grasp some kind of information out of that. But if we're able to actually represent that data in a way that is not only useful to us as scientists, but also to people just in the general population, can actually look at this and see, okay, we're looking at, you know, the momentum that's going through here, the density of various things, then we can actually see an idea of what's happening here. And clearly there's a lot of things going on behind the scenes with this. We've chosen what specifically to show in this representation. we might have another set of data from the same experiments that'll show us something else completely different. But the goal is the same. We're looking for something within that data that will help us make the discovery we're hoping to make. So visualization can be classified in two different ways. The two big categories are scientific and information visualization. And so they're pretty much, those definitions are self-explanatory. They're pretty much exactly what we imagine them to be. I'll start with information visualization. So this is the kind of thing that we're taught when we're younger in, grade school. We're taught how to create graphs. We're taught how to create plots. We do bar charts, those kind of things. And as we continue on later in high school, potentially college, we look at things like parallel coordinates and tree diagrams and those kind of things. And so we're visualizing some type of data, but it's more to inform the user of whatever it is we're looking at. So that could be, how many tweets have I tweeted in the past hour? How many times have I clicked on, X, Y, and Z on Google within the last month kind of thing, and analyze more so trends rather than some type of potential scientific discovery, which is where scientific visualization comes in. So rather than simply looking at, okay, how many times has this happened, or on what day and what time did people look at something the most, we're looking more at data and representations within a scientific domain. So that's when you see things from chemistry, from biology, those kind of things, physics, astrophysics, where we're looking at a scientific domain and data and experiments within those domains themselves. So we can use information visualization to help us with our scientific experiments and whatnot. But the scientific visualization side of things is really where we're looking at the representation of data from our scientific experiments. And so those are the two big words, the big distinctions. We here are going to focus primarily on scientific visualization. And that's what we're going to spend the webinar discussing and showing you guys examples of how to do that. So like I said, all different kinds of ways to visualize things. So again, over here on the right in information visualization, we've got parallel coordinates that are actually visualizing something related to science. If you look at the title of that picture, it's iris data and the sepal width and whatnot, sepal length, petal width, petal length. So that is scientifically related, but that's more of something we go out in the field and we measure these kind of things rather than doing some type of computational experiment, actually working in the computer rather than doing actual hands-on experiments. Same thing with the other two pictures we see in there. Just examples of the types of graphs and charts that you can come up with in information visualization. On the other hand, we have scientific visualization, and each of these pictures is showing us something within a scientific realm. For example, the one of the human, we might be looking at some type of neurobiological experiment, some type of chemical reaction within the brain. If we look at, synapses and when people are thinking about something, what part of the brain lights up, that kind of thing. Looking at molecules and chemicals and all that kind of stuff. Actually simulating chemical reactions within a computer and then looking at the output of, say we get a whole bunch of numbers of the outputs of, if we do 100,000 chemical reactions, actually looking at those through a visual representation rather than just looking at the numbers. And so that's a very common theme that you guys are going to see throughout everything we do here, is representation. So just some examples, by no means a comprehensive list of all the possible ways that you can use these types of visualizations. And I'm going to draw your attention to the list under scientific visualization, because several of these we're going to actually talk about quite a bit later on in this webinar. And those would be things like contours and ISO services, excuse me, volumes, streamlines versus glyphs, that kind of thing. And so those are really, I guess I'll say, visualization techniques depending on the dimension of your data. And we'll talk about that a little bit more later on when we talk about actually deciding what to visualize and how to visualize it. So within scientific visualization, we have lots of different ways that we can actually visualize our data. And a big question would be, well, what do I actually use to build my visualizations? Now that I have my data, now that I've run my experiments and my data is sitting here, how do I build a visualization? And the glorious answer is, it depends. And it depends on what it is we're wanting to visualize, and also how we're going to how we want to visualize it. So visualization software, there's two big ones. They're called Visit and ParaView. You guys have probably heard of those in at least some capacity. Those are very, very robust softwares that have, hundreds of people have spent years developing. They've got all the techniques in there on the previous slide that we discussed very briefly, like streamlines, glyphs, volumes. We have the ability to visualize all of those. And we also have the ability to do that in parallel. So if our data is massive, we've got, terabytes and terabytes of data that we want to visualize, we're not going to want to do that on our personal computers. It will just take way too long, an unrealistic amount of time. And these softwares actually provide you the capability to connect to high performance computing hardware and computational nodes and whatnot, and be able to utilize that to build your visualizations in parallel. And we'll talk a little bit more about that too. And I'll actually show you guys how to set up Visit specifically to work with Comet, since you guys now have access to that out at SDSC. The second way would be libraries. You guys have probably heard these before through R or ggplot2, matplotlib, all those kind of things. But a couple distinctions I'll make for you guys between them beyond what I already said. Things like Visit and ParaView are primarily for scientific visualization. That's what they were built for, and that's what they're primarily used for. You do have the capability to, slice through your data, build graphs based on the information you're looking at one specific point in your data, but that's not necessarily the primary function, depending on what you're trying to do. The libraries, on the other hand, certain ones can be used for both information and scientific visualizations. And something that I'll put out there for you guys to think about would be using them to show information and do information visualizations of scientific data. So I'll let you guys think about that one for a little bit, and if we have questions at the end, we can discuss it more. Visit and ParaView generally don't require too much coding. You do have the ability to generate Python scripts from the steps that you walk through within the GUI. Each one has a pretty well set up GUI that you can use, but you can also set it up to record and build a Python script based on the steps that you walk through to build your visualization. Or you have the ability to generate your own Python script on the outside and bring that into Visit or ParaView and use that to build your visualizations themselves. Things like matplotlib, ggplot, those kind of things generally require a lot more coding. So you do need to know the ins and the outs of whatever library you are using in order to be able to build a meaningful visualization. Whereas with Visit and ParaView, there are unique things to each one, but generally the techniques to visualize are standard, such as, like we talked about, volumes, glyphs, color maps, contours, that kind of thing. Those ways to visualize things are standard, but the way that each one does it might be a little bit different. And we'll go into details about each of those types of visualization in a little bit if you guys don't know what those mean. So I threw out being able to visualize data with high performance computing if you have massive amounts of data. But something that probably a lot of us are looking to do would be actually visualizing data from high performance computing. So if we run all our simulations in parallel, we've got massive amounts of data sitting there that we don't know what to do with. And we want to build a visualization out of that. That's probably the most common way that these softwares will be used. They do allow you to do it locally on your machine, which is probably, at least from what I've seen, the way most people will do it. Because while the experiments are extremely computationally intensive and take a lot of compute power, take a very, very long time to run, and require more than your personal laptop can give it, the data that you actually write out or end up with, you can usually do a visualization from that locally. But there are times where you have an extreme amount of data that just is not feasible to run on your personal laptop, or the data makes sense to be able to split up into multiple different parts and visualize those in parallel as well. So I do like these images. They give a pretty good explanation of how you can actually visualize your data with high performance computing. So the first one here of the human skull, we see how it's the very top line, the blue, green, yellow, and red. Each of those components, or I guess quarters of the skull, are built completely disjoint. They're completely separate. So we can do, if we were going to do it on our laptop, for example, we could do each one individually at a different time. Or if we're running with HPC resources, we can do all four in parallel. We can then actually put those pieces together. So sort of like a puzzle where we put the blue and the green together, the yellow and the red together, and then we can finally put all of those puzzle pieces together into our final visualization. Another way that the data can be split up, which you see in this other image, would be by component. So I'm guessing this is a rocket, some type of aircraft. Each piece is built on a different processor, a different core in parallel to build the visualization. So that's where the different colors come in. Would be each one is being built on a disjoint piece of hardware, a different place. And then each of those can all be brought together into the final image or the final visualization. But something that you do need to keep in mind there is it's almost like running another set of simulations or another set of examples, because we need to be able to figure out, or at least guesstimate, you know, how many nodes am I going to want to run on? How long do I think it's going to take? Those kind of questions will reoccur here, rather than if we just visualize data locally, it's going to do it in serial. So we don't need to worry about that as well. So now I'm going to switch over to Visit and show you guys a few examples so that you can actually see some of these techniques that I'm talking about here. So I guess, Pooja, if there are any questions while I visit.
Yes, there is a question. So someone's asking, what is a glyph?
Yeah, okay. So I'll see if I can find the example in here while I'm opening everything. But essentially, a glyph is a way to show some type of direction within your data, or a velocity. So if we're going to show the way wind is moving in a hurricane, for example, they're usually going to show up, you'll see them as like little arrows Occasionally they'll be colored depending on, you know, like the temperature of the air, that kind of thing. But just think of it as if you're analyzing like a hurricane or a tornado or something, and all the little arrows that are pointing in different directions. Those are considered glyphs. And I actually have another example in a slide after the demo that I'll show you guys, so that you can see what I'm talking about and give you guys a tangible explanation.
Okay, thank you.
Yep. All right. So, can you guys see this?
Yes, it's there on the screen.
Okay, perfect. All right. So this is visit. And I'm going to point out a couple different things while you guys are first starting out. If you guys do have visit on your personal laptops, please feel free to follow along. Or all of these directions are in the document that Wendy sent out to you guys along with the link to the webinar. So you guys can follow along with that. And like we said, we'll be here at the end to answer questions, or you can feel free to email us as well. So if you guys are opening up Visit, you'll notice that there are two different windows, two disjoint things. The first one over here is sort of the control menu. So this is where we'll actually open our data, add in the different types of plots that we've talked about. If we want to do some of the information visualization techniques, looking at, you know, graphs over time, that kind of thing. This is where we're actually going to do that. The other window is just going to be a white screen right now, and that is intentional. So you haven't done anything wrong. That is on purpose. And the reason why you see a blank screen, and you'll see this even after you open a source of data. So I'm going to go ahead and open something real quick so you guys can actually see what I'm talking about. Let's see, I'll open this one. All right. So I opened it, and it clearly tells me it's an active source. There is data there, but nothing shows up in my window. And that is on purpose, because a lot of times, even if you are running locally and running serially on your personal laptop, it is still a computationally intensive operation to actually visualize. And if it tries to keep up with every small change that you make in your data as you're making them, it can overload your computer. It'll add up very, very quickly. So Visit is designed to not actually draw anything or create a visualization that you can see until you click this draw button under the plots. So you see add, operators, delete, hide and show, and then draw. Until we click the draw button, nothing will happen. So just to show you guys something simple, and this is a point that I'm going to talk about in a little bit, of knowing more about your data than you would expect to need to. But I know this data set, I know I want to add in pseudo color, so a color map type of thing based on this variable within the data called Hardy Global. So you see it mentions it down here, tells you exactly what's here. It's going to create a pseudo color plot of this variable. And you see draw went from grayed out to actually being colored. If I click draw, there is my plot. So something actually comes up once I hit draw. Are there any questions, Pooja?
No, none so far.
Okay, perfect. So something I'd like to point out to you guys as well is if I try to do this exact same steps to something else, it will not work. Or I'll say chances are it will not work. So I'm going to bring up, let's see. I'm going to first clear this data, and I'm going to open up, we'll do this one. So another completely different data set that we're going to look at. I'm going to try and add in the exact same plot. And if we look under pseudocolor, we see there is nothing that we can actually map colors to, or a color map. So that leaves us with the question, what do I actually use for the data that I'm attempting to visualize? And again, it's that wonderful answer of it depends. So for this specific data, there can be other things that we actually look at. If we have vectors, let's try this. Yeah, so to answer the question about glyphs, these are a type of glyph. They're the vectors that we're looking at in this specific data set. So we see that each of the arrows is associated with a color, and then also it is pointing in different directions. So it's giving us 2 dimensions of data within here. So the color, as we see over here on the color map, is mapped to velocity. And we saw that when we came over here and actually looked at vectors, this variable was down here, VEL, that's velocity. And that is how the people who did these experiments and created this data set actually set it up to output this data so that we can visualize it. They knew ahead of time, I want to actually look at the velocity of whatever is within this data set, so that's what we're going to output. Whereas if we came down here and tried to put in volumes, or I'm going to come up here and look at contours. Contours are more a one-dimensional type of data. So while there might be something in there that we can create, we won't be able to create a vector or a glyph from the data output specifically to be used for contours. So yeah, just to give you guys can see kind of the spiral in here. So for whoever asked what glyphs were, this is an example of a glyph. And there are a lot of different subsets of glyphs, but we're not going to get into that because that could easily become, 2 or 3 semesters worth of scientific visualization courses. So something else that I put in the directions for you guys so that you have the handouts, but I'm going to walk through real quick just so you guys can see how it's done, is actually setting up your version of Visit to be able to link to high performance computing resources. And so that's why we gave you guys access to if you guys were here last week for the Intro to HPC webinar, you saw more of Stampede 2 and what Stampede 2 can actually be used for, and some of the commands for that. But we wanted to show you guys that you can do these kind of things on multiple different machines. It's not specific to one machine and one machine only. The commands and modules available to you might be different from architecture to architecture, but overall, actually setting these things up in Visit are very similar depending on the resources that you're going to use. So I'll just walk you guys through setting up what's called a host profile. And so if we come up here to options and then host profiles, this is where we're actually going to be able to say, I want to connect to Comet, I want to connect to Stampede 2, those kind of things. So if we click on that, we come in here, and as you can see, I've got a whole bunch of them that I'm able to connect to. But chances are, if you guys have never used Visit before and haven't specifically set up these profiles, your hosts window over here will be blank. And that's as expected because you guys haven't actually set it up to associate with any other resources available to you. So if you come down here and click New Host, I'm going to show you guys within here so you guys can actually see the names of things. So first is the host nickname. That's just to give you the name so that you know exactly what you're connecting to within your hosts. Because as you can see on here, I've got all kinds of them that I'm connected to or can connect to. So it's pretty standard procedure to say the location of wherever the supercomputer is. So for example, NERSC is out at Lawrence Berkeley National Laboratory. So that's where it's located, and then the specific supercomputer that we're going to connect to. So in this case, that's Comet. The host remote name, so this is, if you do your username at comet.sdsc.edu, when you're SSHing, the comet.sdsc.edu is the remote host name. So that's what you guys are actually going to want in this little window here. If we come down here to account, it's very important that you use your username associated with Comet. I know for me, for example, depending on the Exceed resource that I'm using, whether it's, Stampede 2, Comet, Bridges out at Pittsburgh, my usernames are actually different because when I set up those accounts, I set up the accounts individually rather than just setting up one account through Exceed. I believe you guys are set up just through Exceed, so your usernames should all be the same. But for me, it's Colleen H. So I put that in the username, and then we'll jump down to connections. Make sure that this is checked. The tunnel data connections through SSH. That should be checked off because that will allow you to actually connect by way of SSHing into the resources that you're wanting. Yeah, so on the launch profiles up here, you got two options. You got host settings, which is where we just filled in all of that. And then we've got launch profiles. If we click on new profile, the profile name down here is going to change and add something there. What we want it to be is normal. I've already got one set to normal, so I don't know if this will yell at me for trying to replicate, but just to show you guys what you need to set up. If we come over here to parallel, if we click launch parallel engine, these are the types of settings that you'll want to use, similar to how if you request an interactive job through the terminal on Comet, for example, you'll be asked, how many nodes do you want? How long do you want them? What account should those be charged to? Do we want to use the regular queue, the debug, the premium, those kind of things? And if you guys don't know what those, buzzwords mean, the Comet documentation at SDSC on their webpage is actually very thorough and very good, and will provide you guys with explanations of all of those things. And we can choose which of these things we actually want to set in our profile. So if we want to set the regular queue, we'll come in here and type in regular after we check this box. If we know we're going to be doing just simple visualizations, but we want to do it on a compute node on Comet, we can say, I want one node, and let's say, I believe they have 16 processors. So we want all the processors on a node. So we can change these things. If I say I want 1 hour, these are the kind of things that will be set as defaults now. So we're setting our default settings. So if we go in and say, I want to connect to Comet, I don't want to change any settings, these are the settings that it will use. Looks like we've got everything there. So what you'll do is click apply, and that will set everything. the final step will be come up here, back up to options in your menu bar. And at the very bottom, there's this option that says Save Settings. And so you're actually going to want to click that. Otherwise, the changes that you've made won't be saved once we close this window. But since we've actually set everything up, we've clicked Save the Settings, I can actually click Dismiss. And so the visualizations that we just did right here, we can actually do on Comet as well. You guys are free to try that. The instructions are all in the document that we provided to you, very detailed so that you don't miss any steps. There are pictures included to show you guys what you should see. I'm gonna clear this out and bring up the first example that I showed you. So you guys have all of this data available to you as well if you downloaded the data folder from the website. I'm going to open this. I'm not connected to Comet at the moment, but if I say I want to draw my pseudo color of this variable, and then I click draw, and I'm not going to do it at the moment because I'm going to explain what happens. If we've said that I want to be connected to Comet, which is what we just walked through, and I click draw, that is when it's actually going to go and request computing resources from Comet for you to be able to run on. So when I click draw, you guys should see a little screen pop up that tells you that it's waiting for the connection and then also waiting for the resources that you asked for. You do have the option then to change the default settings. So like I said earlier, we just set it to one node, but let's say we're going to need to build a massive visualization that's going to take a while and we want maybe 10 nodes. That'll give you the option at that point to actually do it. So when you click draw, that comes up. Once your resources come back, you'll actually see the image that you had set up to draw. So I'm going to switch back to my slides. And while I do that, Pooja, if there are any questions.
Yes, there's a bunch of questions. So to begin with, someone's asking, what is the data type that Visit can handle, and how do you pre-process your data for Visit?
Yeah, so can you say just the first one again?
Yes, please. What type of data types can Visit handle?
Okay, so there are actually, I want to say probably 150, 200 different data types that Visit can handle. I actually have the list right here. Let me pull that up. If I can find it. But that's actually a good question, and I didn't touch on that. And I apologize for not mentioning that. Let's see. Yeah, so there's probably about 150 or so things on here. And so some of the most common ones, if you noticed in the examples that I was going through, the primary example of what was shown is called a .silo file. That's a very common one. Another common one would be a .bov file, a .obj, those kind of things are the most common ones, but certain types of data and certain simulations that you're running can also utilize other types. So I know something common in the astrophysics world and astronomy is called a .enzo file, for example. So let's see. Yeah, so I don't want to keep throwing out the answer of it depends, but a lot of the examples and smaller things that have similar like commonalities between them can use those common types. And if you go to I believe it's if you type in Google visit users, the visit website should pop up, and on there it'll provide you the big list of everything that it can actually handle in visit. If you develop something and it can't handle the type, oftentimes you can convert it on the side and bring it in as a type that visit can actually handle, but it will give you errors if it's something that visit doesn't recognize.
Okay, so the next question was, how is the data prepared?
Okay, yeah, that's a great question. And that's something that, let me see. Okay, so that kind of goes along with this slide that I was going to talk about next briefly. A lot of times what you can do when you're running your experiments, there's a lot of preparation that goes into thinking about what types of visualizations you're going to want to do, what you're going to want to visualize. And so there are packages out there that, can output, for example, like a .obj file or a .bov file, but you also have the option of simply writing your own functions to do that. So if I want to output a .obj file, there are libraries out there to be able to do that. Or if I'm doing something very specific to my data, I can actually write my own functions to say, okay, I want to pull out velocity and magnitude, and I'm going to write those to a file. So each file type has different formats, so you'll have to look those up because each one is very specific to whatever you're doing. But many times it's simply, if I've got 3 different types of files that can work for whatever I'm trying to do, it's simply just making the choice of whatever you're partial to or whatever your preference is.
Okay, so there is another question. So someone's saying that there are some differences between the instructions that you gave in the PDF and the stuff that you showed on screen with respect to the parallel visit. And more specifically, they're saying that PDF has parallel launch window Q sub MPI run, but it doesn't seem to be there in your instructions.
Okay, it is possible that I skipped that step, and if I did, I apologize. You should have MPI run set when you're setting up Comet. That should be checked off and set to MPI run. And if you do have questions about that, feel free to stick around afterwards, and I will double check those instructions and run through them again and make sure that they are correct. So I do apologize that I skipped that step.
Okay, so another question was with regards to choosing a software. So how is ParaView different than Visit?
So they are very similar. A lot of that, again, comes down to your personal preference. So the way that some stuff is done in the background between the two is done differently, but ultimately you will come up with very similar answers. For me personally, I started with Visit when I was learning how to do these things. So that's just what I've stuck with. But I know a lot of people that use ParaView and are able to come up with, very similar visualizations, go through very similar processes. You can do Python scripting for both. And you can also do, and we'll talk about this in a sec, but like batch jobs, connecting to HPC resources, the steps that you walk through to do so just might be a little bit different.
Okay. So there's a couple of other questions, but if you have something that you want to talk about, we can always take them in Q&A session.
Okay, yeah, I've just got a couple more points I want to make. These won't be by any means the holy grail of answers for these kind of things. A lot of it is going to come down to practice. So the question that asked, how do I know what to visualize, or how do I actually set things up to visualize, is a fantastic question. And for me personally, I'm still learning that. It comes down to the type of data you're doing, what you're wanting to visualize, the experiments. But these are going to be just sort of an outline for you guys when you're thinking about what it is that you're trying to set up. So I mentioned vector operations, and I showed you guys that a little bit. But there are three main types of operations that you can do: scalar, vector, and volume. Scalar will be your like one-dimensional plots. So when we did the color exercise at the very beginning, looking for numbers greater than 70, that was scalar. And similar to this picture in the far left corner down here. Vectors is in the middle. Those are your glyphs, streamlines, those kind of things that are two-dimensional. So we can show, for example, temperature of the air or temperature of water, and also the direction that it's flowing. Words that'll come up a lot with vectors are velocity and magnitude when you're actually trying to set those kind of things up. And then volume is your three-dimensional. A very common example with that is looking at like the human brain or the human skull. Or I guess in general, even just the human body, those kind of things, where you can actually set up a three-dimensional segment of data. So like we see down here, we've got the human brain from all different sides. That full volume is there based on the data that we're visualizing. And then we can actually look at multiple different aspects within that data, depending on what we've set up. So looks like the color map here is functional connectivity strength, where blue is very low and red is very high. And we can move around and maneuver throughout the volume to look at all aspects of it and all sides. So those are by no means hard, fast rules, but just some guidelines to help you guys sort of narrow down how to visualize various types of data if you're handed data from somebody else and aren't sure what to do with it. So we've talked about most of these. How do I visualize it? What routes can I go to actually visualize my data? Locally, what I refer to here as interactively is what we just walked through, going step by step through actually bringing in the data, setting up the operations we wanted to do, and then actually drawing what we said to draw. You can also set those up, as I mentioned, in Python scripts. You can set Visit up to actually record a Python script for you based on what you do, or you can write your own from the outside. And the same idea on HPC resources, you can run it interactively through Python scripts again, or you can actually set up a batch job. And Virginia talked about that a little bit last week, where you set up a script, so you tell it how many nodes you want, how long you want them, and then walk through the different steps that you want to go through to actually visualize. Depending on the resources that you're using, the version of Visit or ParaView might be different, so you'll need to check with the documentation for each of those specific resources that you're using. But with the difference between doing it step by step interactively and the batch job is we write everything out in advance in a batch job, and then we send that off into the queue and we wait. And once it comes back, then we'll actually have our visual that you can set up either, you know, if it's an animation, you can set it up as a movie, or if it's just an image, you can set it to like a JPEG, PNG, anything like that. And there are examples of batch scripts in the tutorial data that you guys downloaded previously. So those, I think, are just some final thoughts. We'll just finish up with a quick summary, and then we can take a few more questions during the 15-minute question time. So you guys have asked a lot of these questions already, and I love that you guys are thinking so far ahead. So you guys clearly have ideas in mind of what to visualize and how to go about it. So that really excites me, because that's, in my opinion, a very fun aspect of actually running simulations. So same ideas here, just reiterate, we want to plan ahead when we're actually writing our code to run our experiments and run our simulations, to be able to output data of what we want to visualize. So like I've said, if you want to actually visualize the direction of wind, for example, we want to be able to pull out direction and write that to a file. And that goes with the second point here, that oftentimes what you're visualizing will be custom and tailored to your data. There is no one end-all be-all way to visualize all data. The steps that I showed you guys for one example didn't work for the second example. And so that goes back to knowing something about your data and understanding what's happening behind the scenes. So that's the third point, just understanding your data inside and out. And oftentimes it requires far more understanding of your data than you actually realized. Because once you get down into the nitty-gritty details of doing your visualization, something might come up that you haven't thought about previously, that you should have actually either tested, simulated, whatnot. And then knowing just what methods of visualization are out there, so that you know what resources you can use to visualize whatever data you're doing. And that's what this webinar was targeted at actually equipping you with, giving you guys an overview of all the tools and techniques available, so that you guys have the knowledge and information that you need to be able to not only visualize something, but also ask questions intelligently about how to go about visualizing specific data. And with that, this is my contact info. If you have questions for me specifically, please feel free to reach out. Otherwise, feel free to use the info at SH Institute email if you have general questions. Otherwise, I think I've exhausted everything I have, so we can, I'll turn it back over to Pooja for any logistics at the very end.
Thank you, Colleen. That was a great talk. So before we start the Q&A session, I would again like to request everyone to please fill out that form. I think Wendy put out the link to the form in the chat, and she'll put it out again. But please do fill that out. It's only going to take you 2 minutes, I promise, but it's very important for us. So I do request everyone to fill that out. So we'll start our Q&A session now, and we'll cover some of the questions that we couldn't get to during the webinar. So one of the questions that was being asked was, do we need to request an allocation to run on Comet?
Beyond this webinar, yes, you will. We provided you the access for this webinar specifically, and somebody else can correct me if I'm wrong, but I believe you have access for 30 days after this webinar. Beyond those 30 days, you will need to request your own allocations.
Okay. And then there is another question. So I think this one is referring to one of the images that you showed. I think it was the second or third slide before the last slide. So, and they're saying, are those glyphs in the middle image the wind?
This one?
Yeah, I think that would.
Yes, so in this image specifically, the color refers to the speed of the wind, with red being faster, blue being slower, and then the way that the glyphs point is the direction of the wind. So this image is actually showing, if you look down towards where Florida is in the US, it's actually a hurricane.
Okay. So now there is a question about what if you have large data set? So someone's asking, if I have a 10 terabyte data set that I want to visualize, do I upload the data set to the host system that is Comet before processing, before visualizing with Visit?
Yes. So whatever system you choose to use to visualize, so for example, here, Comet, you can actually upload your data to Comet so that you don't have to store that somehow locally, because that is a lot of data. And then through Visit, I didn't actually do it, but if you tell it, I want to use Comet resources, rather than where I went through my local hard drive to choose files, it will actually let you go through your directories on Comet and choose the data through the GUI. So rather than doing it through the terminal, it'll give you the option to see and choose what files you want to on Comet there.
Okay. Is there a document for choosing the plot type versus data type or dimension? So I guess they're asking, how do you choose what kind of plot you want to do? Maybe something like a pseudo color versus an isosurface versus something else.
Mm-hmm. Yeah. So that, I guess there's a couple ways you can go about deciding. One would be if you have in mind what specifically you want to visualize. And there are some great resources out there to help decide between, one visualization technique versus another if they are similar. So I will actually, I'll create or I guess compile a list of a couple of those that I use personally all the time, and make sure that everybody has access to them so that they have the same resources available. But yeah, without knowing too much about their data or what they're wanting to visualize, I can only give a general answer at this point.
Okay. Then one of the question is, do Visit and ParaView support real-time visualization?
To a certain extent, yes. Depending on how large your data is, and what you're trying to do, there are some plugins and whatnot available to do what's called in situ visualization. And that's a whole other discussion and topic. But they do try, I believe, from what I've seen, to shy away from doing real time just because it is computationally intensive. But I believe there is some way you can turn that on, but off the top of my head, I'm not sure what it is.
Okay. Well, so the last question is what someone had asked before, which is differences between what you demonstrated and what was possibly there in the instruction sheet. So I think if you can,
Yeah, let's pull that up.
what you're doing. Right, and I think Andy is the person who was asking that question. So Andy, Colleen is going to show you what she did, and if you want, you can use the raise your hand question and actually ask your question yourself.
Yeah, so let me find. Alrighty. All right. So, I believe the step I accidentally skipped was setting this parallel launch method. And so for this one, that would be, oh, let's see, Q sub MPI run. Nope, just MPI run, sorry. So depending on the system, this will actually be set differently. And you can determine which one is for whatever system you're using through the documentation of those website or of those systems, sorry, on their websites. Let me find. Let me pull up the document that I'd written up. Yeah, so I'll go ahead and show you guys this. Yeah, so if we go host profiles, one thing I did skip was the path to visit installation. And so for comment, that is /opt/visit. So let's go in here and let's see. This one. I believe that will be blank. So you will need to put the /opt/visit. You can copy and paste it directly from the documentation to make sure you guys don't have any errors there. But let's just walk through this, make sure I didn't skip anything else. Ah, yes, it is supposed to be Q sub MPI run. All right, so Q sub MPI run. And Q sub is how you would actually submit a job interactively. If you're going to request, 2 nodes for an hour, you would use the Q sub command. And then for this system, it's actually called normal, not regular. So that's just your typical queue. Nothing like priority or anything like that.
Okay. I don't know if you can hear me, but this is actually Andy who was asking the question. Yeah, I've got normal in there and everything following the PDF. And I can even SSH into Comet for my normal SSH program.
Mhm.
But then when I try to open that host profile with visit, I go to open, select instead of local, I select the host, the SC comment.
Mhm.
It kind of hangs for a little bit, and then finally a window pops up, and then it asks me for my password. I put that in, and then it just sits there with a metadata server launch progress window, and says visit is waiting for a metadata server to launch on Comet.
Yes.
And then I don't know how long that typically takes to complete, but or if that just depends upon how busy the system is at the time.
Yeah, so
But
that's more on their side.
Okay.
And sometimes, sorry, I didn't mean to cut you off, but sometimes you might want to try decreasing your time limit. I know a lot of times a 30-minute time limit will get through a lot faster, but yeah, I have seen them hang for a couple hours at sometimes.
Okay, so it's not necessarily something I have wrong with my settings.
No. If you have everything set with the settings in the document, and if it's popping up saying it's waiting, it's waiting on visit on the HPC resource side. It's not waiting on you.
Okay. That answers my question. Thank you.
Awesome. No problem.
Right. There is another question about, there is another request actually. Could you please demonstrate how a batch file can be submitted to Comet HPC system? I can see an example file in batch visit folder.
Yeah, so on Comet, if you're logged in through the terminal, you'll want to make sure that whatever batch file you have that you're wanting to submit is on Comet. But once you have it there, you can type sbatch space the name of your file and hit enter. And if you've done it correct, like if there's no errors in your file that it can find, then it will kick back what's called a job ID. It's usually a long string of numbers. And that means that your batch job has successfully been submitted and you're now just waiting.
Okay.
We can probably do one more, Pooja, if there's another. Otherwise, I think our 15 minutes are up.
So I don't think there are any further questions at the moment. I think that was the last question.
Okay, perfect. Like I said, my contact info is in the slides that we'll make available. So feel free to reach out to us through the SHI email or my personal email. I'm happy to answer any questions I can. Otherwise, Pooja, if there's any other logistics.
Right. The only logistics is, guys, fill out that form. You're probably tired of hearing that from me, but there is a reason I'm saying it again and again. But yeah, other than that, please do let us know if there is any topics that you would like to have our future webinars on, or if you would like a follow-up of a visualization webinar or something else. So do let us know. Thank you so much.
Thank you, guys. We appreciate you taking the time to join us.